are going to see the synthesis and release of thyroid hormone. We have the thyroid gland. The thyroid gland is made of follicular cells. These are the follicular cells, and these cells surround a follicle. This is one follicle, and we have many follicles. This is called the colloid. The colloid weighs like 800 micrograms, and of those 800, 600 contain the T4, T3, thyroglobulin, etc. Next, these cells would have the basolateral phase, that is this touching the blood, and this is the apical cell, the apical phase. This apical side of the membrane is the one that is touching the colloid, and here in the colloid is where T4 and T3 is going to be first, or when the ionization, organification, coupling happens here, then it will be returned to the cell and will be cleavage, and T4 and T3 will be released. We will see these steps in details. So first, we need iodine. So the iodine is going to enter from the blood to the cell here in the basolateral membrane due to a symporter of sodium and iodine. Next, iodine is here. Alright. The iodine is going to be transported here through the pendrin. Pendrin is the enzyme that put it here in the colloid. Now, this iodine is going to undergo oxidation. The oxidation is accomplished by the thyroperoxidase enzyme. So we would convert this iodine to molecular iodine. And this is happening here in the apical membrane. This was the oxidation. Next, there is organification. In organification, the iodine is going to be bound to thyroglobulin. These cells produces thyroglobulin. Let's view how this happens. This is a cell, and this is the ribosome. The ribosome translates a protein. This protein is going to be translated, at the beginning here in the cytoplasm, but then goes into the reticulum, the endoplasmic reticulum. So the ribosome stops here, and the synthesis continues here. Next, the protein is going to undergo, endocytosis, exocytosis, transcytosis, all this to the Golgi apparatus, and in the Golgi apparatus there are modifications like glycosylation, etc. And finally, at the end, the thyroglobulin is exocytized here into the colloid. Now here we have the thyroglobulin molecule, protein, and molecular iodine. This thyroglobulin protein has a lot of thyroxine residues, and these thyroxine residues are going to be iodinized with this. This is the organification step, and is also catalyzed by the thyroperoxidase enzyme. So we would have the iodine in the residues of tyrosine, like this, and then the coupling is, we have this is all the protein, etc. Here, let's say we have a residue of tyrosine with one iodine, and here we have another tyrosine with iodine. Actually, in the tyrosine molecule, we can put one or two iodines. So let's say this one has two iodines. The coupling is one tyrosine residue and another tyrosine residue, each one with iodine, could be one or two or any combination. Then they are going to be bound. This would bind to tyrosine with iodines. This is called monoionized tyrosine, and this is the monoionized tyrosine. Together, if we have two iodines and one iodine, we will form T3. And if we have 2 and 2, we will form T4. But T3 and T4 remain bound to thyroglobulin. So it's not still T4 nor T3. This was coupling. It's still catalyzed by the thyroid peroxidase enzyme. Next, now that we have T3 and T4 here, like embedded in the thyroglobulin, this cell is going to do pinocytosis. This pinocytosis is like drinking this colloid fluid with the thyroglobulin and T4 and T3. Next, this thyroglobulin is in a vesicle, and this vesicle is going to be fused with lysosomes. So the lysosomes destroys the thyroglobulin and destroys the binding of T4, T3 to the thyroglobulin. And is that how in this cell, this is the cell now, we have T3 and T4 in a vesicle. This is then going to fuse with the basolateral membrane and releases T3 and T4. In the other hand, the thyroglobulin, yes, is going to be destroyed by the lysosomes, but the iodine that is in the, let's say that here we have another residue of tyrosine with iodine, and here iodines, in two iodines in one tyrosine. These iodines that are not in T3 nor T4, these are tyrosine residues with iodine that were not coupled. So we have here monoionized iodinized tyrosine, and these iodines are going to be recycled. So these iodines are recycled and again are used for next coupling, next organification. Now here in the blood now, T3 and T4 goes to every cell, and the receptor for T3 and T4 is in the nucleus, because these are hydrophobic hormones. So we need there is a protein that transports the T4 and T3. This is mainly the thyroglobulin binding protein. This transports like 70% of the thyroid hormone, but 99% of the thyroid hormone is bound to protein. So the other 29% is bound to thyroglobulin prealbumin, that is transthyretin, and to albumin. The remainder 1% of free T4 and T3, this is the one that is not bound to protein. This remaining T3 and T4 is, the one that regulates the TSH. The TSH is regulated by free hormone, as always in endocrinology. Next, this T4 and T3 is the one that goes into the blood, into the cell. And when the cell is removing this T3 and T4, then the dissociation of the T3 and T4 that is bound to the protein, etc. But the important issue is that most of the thyroid hormone that is produced in the follicles is T4. So we have like 20:1 ratio of T4 to T3. But T3 has much more affinity, like 10 times more affinity to the receptor in the cells. So the more active form is T3. There is peripheral conversion in the tissue from T4 to T3. This is the 5' deionidase. Removes one iodine, and now we have T3. This is also found in the thyroid cells. This deionidase enzyme also patients can have deficiency of this enzyme and will present with some changes in the thyroid function test that we will see all this in the subsequent videos. Also, T4 can be converted to T3R. This is reverse T3, and reverse T3 is just an inactive hormone. It's like a way of inactivate T4 because this is non-functional. This can also be done in any peripheral tissue. The T3, most of the T3 that is produced by the peripheral tissue. This is the thyroid gland releases mostly T4. T4 is converted in the peripheral tissue to T3, and then T3 goes to the cells because there is increased affinity from T3 to the receptor in the cell. This is the T4 is converted to T3, let's say skeletal muscle, liver, etc., and then is released again, and now goes to every cell. And we already talked about the effects of, or if not, we will talk them in the next video with a brief overview of this, the effects of thyroid hormone. Also, the conversion to T4 to T3 is blocked by glucocorticoids, propranolol, and propylturacil. So these will be drugs that we will use in hyperthyroidism. And the glucocorticoids and beta blocker, the propranolol, increases the conversion from T4 to reverse T3. Also, when the T4 and T3 is released here into the blood, there is also, release of thyroglobulin. This thyroglobulin is increased in many diseases, like for example, if we have goiter, if we have some thyroid cancer or thyroiditis, when T4 and T3 be released, let's say the patient has hyperthyroidism, also we will see increase in thyroglobulin, like in the Kerbein thyroiditis, for example. After removal of the thyroid gland, if we see thyroglobulin, that means recurrence of the cancer. I think it's everything for now. In the subsequent video, we will talk about the actions of thyroid hormone.